really about shells. It's really about cool Florida shells. And the reason why I decided to do that is that a couple of projects in the recent, let's say within the past year, have prompted me to begin wrapping my brain around Florida shells instead of only local, Southwest Florida, Sanibel Captiva, regular fare. A lot of what we do now, especially because of the pandemic, is image oriented. We try to reach our audience using videos, images, given that it's hard for people to come here, especially when it comes to visiting the collection. The first part of this talk, I'll introduce a couple of projects that really kind of funneled my interest in becoming more knowledgeable about Florida shells in general. Evidently, the existing effort is the local effort that we've been working on since 1998. It sounds horrible saying that because one is betraying that they're not so young age, but it's our Southwest Florida Shell Guide online that, unbeknownst to you, almost every week or sometimes twice a week, I tweak this resource. I add new images, I correct typos, change names as the species names are updated in worms and in the science world. So this is a project that it now carries about 400 local species from Sanibel Captiva and the neighboring areas. And obviously it's all visually oriented. So, what really helps is looking at the image, comparing to the shell you have in your hand, and that way acting the resource act as an iconography. It's a classic image comparing to the object resource. We were lucky enough to have had 2.5 years of federal support, federal money devoted to the digital imaging project. Here you see a picture up of the two initial members of the project, the workers that made a huge difference, Patricia Starkey and James Kelly, shown here with me in the photo room in the museum. So this project, most of it, right now we have around 4,000 composite images for a total of roughly 16,000 individual shell images, and they are posted in our shell catalog through the museum website. And as I said earlier, most of those are Florida shells. So you can see that I had to be very careful because those images, they come with the species name and they're there for everyone to see. So we want to make sure that they get the best information. On the other hand, having this type of resource open to the public makes it very easy to make changes. So if I have something that's not the way it should be, trust me, I'll hear it in less than 24 hours, and I'll be able to go back through the back door of the programs and make the change that's needed. This is just an example through the Global Biodiversity Information Facility, GBIF, that has those are all our images from the museum collection. And most of the shells you see here, this is just the tip of the iceberg. As I said, we have between 3,000 and 4,000 lots imaged, and most of them are Florida species. Another project that we started last year is a project funded by the National Science Foundation called Mobilizing Millions of Marine Mollusks to the Eastern Seaboard. And this is done in conjunction with 14 other large American collections, mostly from the eastern half of the country. This is actually, by the way, a photo of the Facebook page for the project. So if you haven't liked that, if you're in Facebook, please do so to help us promote the project. And we're trying to normalize, to make standard the nomenclature of species, to make standard geographic coordinates for the lots in museum collections, and so that it becomes a much more powerful resource than those collections already are. And again, with many thanks to the National Science Foundation for the 4-year grant we received together with the other museums. This is just a page of our catalog showing a while ago all the lots that are included in the Eastern Seaboard project, around 20,618 lots. And as again, a definition of the Eastern Seaboard in this case is all American states from Maine through Texas, going through Florida and the Gulf of Mexico. We have some products that are coming out of this project, and mostly with the goal of doing outreach and public education. Thanks to Farsight and the good brains of our new director, Sam Ankerson. Was actually Sam's idea to when he first saw the images, and he turns to me and said, "Do you think the images from the digital imaging project could stand big magnification, let's say to 4 feet wide?" And I said, "Well, let's give it a try." I sent a couple of files for printing, and lo and behold, they came out beautiful. So we embarked on this project, the exhibition In Focus, Precision Photography of Extraordinary and Uncommon Shells, most of which Florida shells, that will be there until late November. So if you haven't been to the museum yet, if you're coming back for the season, make sure you come in and visit. And finally, which will be now the keystone, the driving force behind the rest of my talk, is the poster, Cool Florida Shells. I always like to do posters and play with design and images. Last year, during the lockdown, we were home for about 5 weeks. So I picked up my little laptop and played with that. A few months later, we had a poster. I had to check a couple of names that were not proper, and I know some of the members of our audience today helped me. They saw earlier versions of that and helped me change some of the names and make them right. So what I want to do today is take Cool Florida shells from the poster and just talk about them. And at the end, if you have any comments or any questions, I'll be here to socialize and talk with you. The poster is for sale at the store, $15, and it's 18 width by 24 inches tall. And it's super nice, high resolution. I hope you like it. We debated whether or not to do a poster. Some folks, oh, posters are a thing of the past, the 70s thing, but they're already selling like hotcakes. So it's a good thing we did. So without further ado, let's begin and see what we have to see here. So I'll bring the poster to a little corner. The way we're going to do that, we're going to take images from the poster and I'll expound on them like that. We all know about the queen conch, and as some of you may notice there, the name of the scientific name is superseded. The queen conch is a very valuable commercial species, as some of you know, and it was very common in Florida. It almost disappeared from the East Coast. Back then, the DEP, the Department of Environmental Protection, issued a moratorium on collecting of that, and I think a lot of that is still going on. Difficult to manage the fisheries of this species because most conchs, true conchs in the family Strombidae, they have larvae that last a long time drifting in the water. So the queen conch that you see in Florida may have been born somewhere in Cuba or lower in the Caribbean. So it's very difficult to regulate a species when there is communication of the genetic pool of that species from one country actually will grow somewhere else. The name change is strange. In my days, it used to be Strombus gigas or gigas or gigas or whichever way you like to pronounce that. And for a while, but not for too long, it went from the genus Strombus to the genus Eustrombus, E-U Strombus. And that lasted probably a couple of years, then to Lobatus, which is the name we have in this image. And finally, a couple of years ago, some genetic studies invoked, I believe, the creation of a new genus, Aliger. If you are a collector, you worry about your labels. Do not worry about changing those labels, because the changes you make today will be redone tomorrow. As long as you have the correct species name, that's what matters. Next one, it's a favorite of many of us. The whole group is super cool, and this is a Caribbean carrier shell. They belong to this genus Onustus, which is a group of carrier shells that carry things, but they don't carry very large shells. If you come here to the museum and look at the exhibit on carrier shells, you notice that some of them can carry shells that are almost as large as their own shell. But in this case, the Caribbean carrier shell and the longlii, the other species, deeper water species, they carry usually small bivalve valves or small pebbles. But yet, it's a spectacular species. I love the way the base looks like, very shiny in some areas. It can come in different colors. So this is your Caribbean carrier snail, family Xenophoridae. Again, very common species in the Caribbean and parts of the East Coast, extremely confusing with our own Florida fighting conch. The West Indies fighting conch, shown here with the operculum, is a species that some suspect may hybridize with the Florida fighting conch. And in some cases, there are areas in Florida where you are not able to distinguish or really tell, no, this is A or this is B. You know, it's very hard to separate. In this case, there's no question. And usually, you know, one of the first things we check to be able to separate the species is whether or not there is a row of strong spines / nodules in the last whorl, being that the Florida fighting conch usually doesn't have that. And the spire also tends to be more elongate in the West Indies fighting conch, but not always. So hence all the confusion. Next, another relative, same family. And we have our hawkwing, the hawkwing conch, which in parts of its range come as what we call, effectively, we call them the dwarf. Um, the dwarf hawkwing conchs, in some cases, you know, just a little bigger than an inch in size. And we consider them to be dwarfs because they have small size and yet they are reproductively viable, meaning they can reproduce at that small size. This one is from Missouri Key in the Florida Keys. And again, we try to show the operculum. And we may have noticed all the images in our digital imaging project have a scale line in millimeters and any forms of original labels that came from the old collections that the shells come from. We do not put our own label, as all the information is already in the catalog that you're looking at when you're looking online. This one here is a very super elegant species in the genus Favartia, a species named by Ed Petuk, who was for many years at Florida Atlantic University, who has named a lot of Florida species. This was named after Bob Pace of Miami, Florida. And it is a member of the genus, as I said, Favartia, which is a genus of small muricids. They're super elegant. They have this structure that looks like honeycomb in the shell sculpture. So this one collected in Marathon back, I must say, sometime back in the day. Well, next one is a favorite also of the local crowd, and why not? I mean, it's really something else. Image taken by James Kelly. And as with all the images done by James, it's just fantastic. I mean, I love the work he's done. There are no hard shadows. There are no bright spots. If you look at those shell images, the external views on the left, the internal views, a view on the top of the shell, and here the little mesoplex. As many of you know, I'm sure, the angel wing, the members of the pair of valves, they do not touch each other. The animal actually makes all the connection between one valve and the next one, helped by a couple of structures, one of them being the mesoplex. This mesoplex is inserted in the muscles of the animal and plays a role in the digging efforts of that bivalve that moves up and down a hole, living in that hole for its entire life, as you know. So also I want to call attention that we have some super important historical collections here in the museum. The main one, the first one we received was still before my days here. Tucker, our founding director, Tucker, was very close to Dale Stingley, who was a professor actually at the, I think, the Northeastern University in Chicago. And he moved to Alva and he started collecting here way back. So this lot here was collected on Captiva by him, by Dale V. Stingley in 1948. Next, a species that we don't have on our shores, but it's always a hit, the Atlantic trumpet triton, which is the counterpart to the Pacific trumpet triton, and relatively common species in the tropics and in our Western Atlantic side of the ocean. One of the largest species in this ocean. It's smaller than its Pacific sister. It is here at the museum where you will find the world record shell for that species in our worldwide record size shells display. You can see the operculum. One of the main differences between the Pacific and the Atlantic trumpet tritons is that in the Atlantic trumpet trout, you see a set of pairs of little white, quote unquote, teeth along the lip here. Um, this is the Atlantic. The Pacific, there is only one and one and one. They're not pairs. They're just single um, white, quote unquote, teeth. Also, species we do not have here. In this case, a shell that came with a collection of Don Swenson. Some of you who have been to the Florida United Molluscologists may remember Don, and he would bring shells, put them on a table in a little piece of fabric, and let people come and enjoy them. And at some point, he asked me if the museum would be interested in his shells, and the answer was yes. And the main reason why I really wanted to bring those shells to the museum and accept his gift was that he collected over a period of 25 years in 3 beaches only. So he has like this historical record of beaches in Stewart and Palm Beach County on the east coast of Florida. And this green turban shell here, a star snail, I'm sorry, is from Coral Cove, which was the place he had his residence there. And again, another favorite, this one collected by dredging of Fort Myers Beach. It's a really well preserved Cabrites murex. You can see a sign of a well preserved shell is the presence of the protoconch. And a little operculum here, it was live collected in 1964. And this is another species that has jumped from genus to genus. And it used to be Murex haustellum, now under the genus Murex, a genus named as a tribute to Emily Vokes. And this here is another specimen, the same species that was collected back in 2018 during one of the cruises of the University of South Florida that participated on. That one was live collected, and it has this beautiful color that will eventually fade away as the shell age. I would be remiss if I didn't include micromollusks. This is actually not one of the smallest of the micros, but still it's a relatively small species. The scale line here is in millimeters, as I think I mentioned before. This is a columbellid dove snail collected here by Lois Dunham, who was a museum volunteer, collected in 1998. I always like to remind everyone that most of the biodiversity of mollusks is of small size. Most of the mollusks, when you look at the whole thing as a group, most species in terms of percentage are smaller than 1 inch, and many are smaller than 1/2 inch. This one probably was like 1, 2, 3, 4, 5, 6, 7. It's about 9 millimeters, which is a little less than 1/2 inch in size, not totally too small. And again, this here is a species that I put here. Well, it's in the poster as all of the ones I've been showing. But there's something I want to say about the person who brought this species to the museum was Jeff Oates, who many of you knew and who passed away recently. Jeff brought this shell to the museum probably about 10 years ago or more. And it's a shell that you normally do not find. Here is the long spine star snail, Lithopoma amphibium. And Jeff had found that on the east end of Sanibel in the water. I was very skeptical when I first saw that. I said, well, let's wait, Jeff. And a couple of years ago, someone else brought another one, collected on the beach in Sanibel. And at that point, I said, well, let's release that Jeff specimen into the shell guide as a local species now. Once I had 2 of them, I was happy that, it was not a shell that had been dropped by someone by accident. So here he is with a nod to the hat and hoping that Jeff is resting in peace, and with many thanks for all he has done, for all of us. The east coast of Florida and the Keys, unlike the west coast, is very rich in species of tritons. And this is one of them. We used to call it Samathium muricinum, now in the genus Guternium. A couple of things I want to show here. Again, this is a shell from Don Swenson's collection, handpicked by him snorkeling in the clear waters of Palm Beach County. There are 3 views as usual, the same shell. On top of the views, you can see a little brown tip. That's the protoconch, which is what remains of the larval shell of that individual after the individual stops being a free-living planktonic larva to become a bottom-crawling snail. So up to the line here, there's a little line where the brown ends and the white begins. That's the moment where the shell transitioned from a open water, freestyled, quote unquote, life, where it was being carried by the currents and swimming a little bit, down to the moment where it settled to the bottom. Most species of triton in the family is Cymatidae or Cymatidae. They have that kind of long-lasting open water larva. Again, a not uncommon species. We bring some bivalves into the picture, but one that really strikes me as super cool, the American bittersweet in the family Glycymeridae, a genus Glycymeris or Glycymeris. And it's one of the largest species of that genus. There are other species of the family here, about a couple more, but this one is the largest one, growing to be, about 5 inches in length in this dimension here. Next one is a relative of, I know you guessed right, of horseconchs and tulips in the family Fasciolariidae, and it's a hemipolygona carinifera. I do not remember the common name, and this was collected off the panhandle of Panama City in northern Florida. And you see the date here, July 1963, collected by Jim Moore. And Jim Moore was still is a very famous person. He's passed away, but he used to do dredgings and take people and do dredgings and then sell the dredging products by the bushel so people could take them home and sort for micromollusks. So a lot of good materials in this museum and in the Florida Museum of Natural History in Gainesville originated from his efforts collecting mainly in the Gulf of Mexico, the rich diversity of mollusks in the Gulf of Mexico. Since I was a kid, I was fascinated by that, and you can see why. I mean, that's the famous or infamous bleeding tooth, Nerita piloronta, also from the Florida Keys. And the name is the allusion to this red spot here that to some may look like blood in the little teeth. Another thing you can see here is that the original label had an incorrect species name. And as part of our curation job here in the museum, we go ahead and make sure that whatever name was there is actually what it is. And in this case, it was not. But we keep that, hoping that, that mistake is not made again. Not a big deal. Distorsius, it's, I really like the whole family. I mean, the way they look like. And there is a reason for the name Distorsius. The shells are kind of distorted. They look like someone has picked up a triton and gave it a yank and bent it. Someone was mad at that triton. And it looks like a Distorsius. And they are really super cool looking. There is a reason also, I believe, why all the aperture or the opening of the shell has all these projecting teeth. They serve to protect the animal from intruders, I guess. It's a very common trick in land snails. A lot of land snails have very complex apertures loaded with things sticking out and projections, and the Distorsius do the same thing. So this one was collected right off from Fort Myers, probably Fort Myers Beach. Curiously enough, I just received a gift from someone who found one in the water in Fort Myers Beach, which is the first one from our area here. So we'll post that also in our shell guide. Same species. A collection of images of the same shell, the same pair, in different views, also from the Don Swenson collection. Kind of iconic Florida species. For those of you who scuba dive, is a species that's usually found on shipwrecks and sunken metal structures. They like to settle on sunken ships and boats and all that. So this species for many years was known as Spondylus ictericus. Someone at some point within the past 10 years found that there was an older name that had been given to the species and not used. So in that case, that species name goes back to the oldest name, provided everything else is okay. The next one is one of my favorite murexes, and it's not super elegant, but it has a Baroque look to it. Is the Paz murex, Paziella pazi or pazai. It's very common in the Gulf of Mexico in deep water. Not common on the beach. You'll never find that on the beach. It's a deep water species. This one collected off the dry tortugas. I was in a cruise, I'll mention a little at the end. I was in a cruise with Dr. Greg Herbert from University of South Florida last weekend. We collected a couple of them again in the same general area. It's a shell that is well known for its very long spines. A species that it's found locally here, the Tampa Bay drill, the Tampa drill. Not common, and for many years I couldn't find it here. And all of a sudden, it's been found by a couple of different people around Bunch Beach. And it looks like those things can come and go depending on the environment, or maybe we just didn't look properly, but it's not very common locally. And it's similar to the Gulf oyster drill to some extent. They all belong in that group of small murexes without spines. Really cool looking when you look carefully. And this was collected on Tampa Bay back in 1962. The frog snails or frog shells, this one just moved from the genus Bursa to something else, and I forgot. I just saw it last weekend. We have to go back now and try to change that. Again, I strongly recommend that you guys who have nice collections do not worry making those changes all the time. As long as you know that the species is that species, because those are bound to keep bouncing until they really resolve the genetics of that. Our collection catalog now is linked to a resource called WORMS, which is the World Register of Marine Species. So our catalog, under each lot now, the link exists that we don't need to go to WORMS. The catalog goes to WORMS and finds the most recent name. And they ask if us, the curators and the collection workers, if we want to accept that change. And if we're okay with that, we just check mark, click a check mark, and the name changes in the collection. So that's the future. So we don't have to worry about making that change all the time. And now a little bivalve, a lister Venus clam, a Periglypta listeri from the Keys, from Isla Morada. And this family, the Venus clam family, Veneri, is the most species-rich bivalve family in the world, as far as we know. There are more than 800 species worldwide. And here alone on Sanibel, we have about 20 species of Venus clams. So it's some of them are really small. Some of them are as large as the southern quahog that can grow to be 7 inches in size, which is pretty large. This is one of the species that's not common locally, if present at all. I don't think they live here. You have to go to the East Coast to find them. Now, this one here is one of the local species. I used to pick them up at Roosevelt Channel between Buckeye and Captiva in very, very fine mud. That mud that actually, if you're not careful, you get stuck to it. It's the green paper mussel. And I also want to make a comment about this collection. Well, it was not donated to us directly. It came from the collection of Alice Anders, who was a very well-known environmentalist, local person here in Sanibel. She donated it to the museum, and it comes from the collection of William C. Brumbach. And Mr. Brumbach, I grew a strong interest in learning more about him, because when I looked at his labels and his material and the dates when he collected, it's super precious, because also, like Stingley's collection, it has a lot of historical value. And as you can see, the date here is March 1964. It's not yesterday. I went online and did some research, and I found that Mr. Brumbach was a member, not a board member, but just a regular member of SCCF in the 60s. And he was a snowbird here in Sanibel in the 60s and the early 70s. And finally, the thing that really, in addition to having donated this collection to the museum indirectly, about 800 lots, and very precious, he also donated a very large collection of pressed plants to the University of South Florida, which is the founding stone, the keystone for their collection of botany in Tampa. So he had this dual natural history interest of collecting shells and collecting plants, Southwest Florida plants. So that's the kind of collection we like to have here. And finally, I want to show you an all-time favorite, your spiny jewel box. This one collected in 1960. This goes to show how we try to show as many views as possible. This composite image is actually a combination of 6 images. So one of them showing the ligament of the shell here, and the collar. And notice how well preserved the collar is. after those many years, basically 60 years after collecting. Another, I photographed that about a week ago in the ship in the middle of the Gulf of Mexico. And that's a live one. And they're showing that beautiful arrangement of spines and such. The Amphiurgus cone collected off the Panhandle, destined. Also, one of the many cone snail species present in Florida waters. This one collected at 85 feet out of Destin. And the flame helmet is the, I wouldn't call the ugly duckling, but is the least known of the three large, the queen, the king being the larger than the flame. Nonetheless, it's a species that's well known from the East Coast and the Florida Keys. And this one is from Pickles Reef, which I believe is near Key West. Glory of the Seas, Venus clam. Always loved it. It has this landscape of small mountain ranges against a white background. Very elegant shell, especially when you collect the pair that's fresh, that still has that very strong brownish red color. This one from, oh, I misspelled Palm Beach, West Palm Beach. So need a change there. This one, your Lady in Waiting, Venus. I always wonder if the looks of the shell has anything to do with the dresses of the lady in waiting characters, but probably. But this is a species that to those of you collecting locally here, it's very well known. It's very common in deeper water in the Gulf of Mexico as well. Finally, one very fine specimen. That's a hard one to beat in terms of preservation of the shell. This one came from the store of Elsie Malone, whose collection is actually at the Sanibel Library on display, very well curated by Jeff Oates and presented to the public at the Sanibel Library. But this specimen is in great shape and was trawled off Sanibel Island. Just want to show a couple of pictures of the work that we did last weekend on a project on biodiversity mollusks with Dr. Greg Herbert, who's shown here. This is Dr. Steve Geiger. I don't know if he's been a speaker with the Shell Club. He's a potential very good speaker for the club too. He works on fisheries and of mollusks, working for the Florida Fish and Wildlife Conservation Commission out of St. Pete, a good friend of mine. And this is Aaron Avery, a very promising doctoral student of Dr. Herbert at USF. I know also, I understand the Shell Club has been helping the University of South Florida as you do for the museum, and I couldn't see a better way of spending your hard-won Shell Show and membership dollars. They really deserve all that. So this is a picture of us the last day of the cruise. This here is a super fancy large screen navigation monitor showing that we're getting back to port in Carabel. I had to show that because those pictures are kind of fresh from the oven, and it still relates to the cool Florida shells to some extent. One of the cool things you see under my legs here is a video ray, and a video ray is like you all know what a drone is. This is the underwater equivalent of a drone. So you send this thing tethered with a line, and it goes up and down. It films whatever you want to film down to a depth of, I believe, 100 feet. So we did a couple of very cool videos during that cruise, showing the dredge coming up with all the animals and all that. I want to thank a couple of sponsors of our museum projects and when it relates to collections, especially. The Institute of Museum Library Services has been funding cataloging and imaging of the collection for the past 6 years. We have the IDIC Bio is the major data aggregator that has been helping us put our collection out in the world with the big dogs. And so is, this is our catalog software, Specify, which is we've been using since for 3 years now, very successfully, very powerful software. The state of Florida, National Science Foundation, Global Biodiversity Information Facility also putting our data out there. They're based in Europe, and our collection data is managed by them also. University of South Florida with efforts going out and looking at living mollusks. Evidently, the Sanibel Captiva Shell Club has been supporting the museum collection efforts and a lot of other educational efforts the museum since before the museum opened. And right now, I'm very grateful that you've been sponsoring annually our subscription to the software. Our collection software is fully sponsored by the Sanibel Shell Club. And last but not least, an anonymous donor that's been helping us with imaging materials and collection materials in the past 18 months. So thank you all for all you're doing. Thank you so much.
Okay, Jose, thank you. That was fantastic. I love your poster. I can't wait to pick up one. And I learned so many great things as always. So thank you for being here. It was terrific.
Yes.
I see a question here from Barbara M. In relation to I mentioned the mesoplex, and by the way, it's a mesoplex is PLAX, and it's the little it looks like a little handlebar of a motorcycle, if you will. And what it does, it doesn't actually attach to the shell. It is a structure that I wish I had an angel wing here. Let's assume that this is an angel wing. You're looking at the angel wing, okay? And that's the inside of the shell. And this is the side that's more rounded. And then there's a side that's more, that tapers off. So it goes in this, it burrows like that. The mesoplex is kind of around here. And what it does, it gives structure to the soft body of the animal. So the muscles can act more efficiently. And one cool thing about all those pholages, the family of the angel wing and the pholad rock borers, is that they burrow by making a rocking. They alternate rocking movements of the shell. That's why the two valves are not attached together. I mean, because if they were attached together, they couldn't do this kind of cool dance they do when they are burrowing into the hard mud. So the mesoplex acts as to give some stiffness to that part of the shell, and to make sure the muscles are where they should be for doing that kind of movement.
Mindy has asked what pictures, she says the pictures are beautiful. What type of camera do you use, Jose?
Yeah, the cameras we use, we tend to use high pixel count, meaning like high definition Nikon bodies, digital DSLRs. And most of the pictures you saw here were taken with Nikon D800s. And they are kind of old fashioned models, like from 2012. And they have, I don't know, for those of you who are into photography, they have a pixel count of, maximum pixel count of 36.2 megapixel, which is pretty high. We also use a Nikon D600, which is 24.2 megapixel. Now, as much as you can do a lot of things now with your cell phones, those images are not cell phone images. I mean, we can blow them out to like 8 feet wide and they still hold. You cannot see the pixels. They are really professional cameras and cool things. And a lot of that is a bunch of the original museum cameras that we no longer have because they became outdated or broke were actually given by the Sanibel Shell Club to begin with more than 20 years ago. So it goes, I mean, the hand-in-hand thing here goes way back. And thank you so much for all you've been doing.
And the other question that came was what kind of lenses you use, or the variety?
Actually, I can show you one. The lenses are, this is a Nikon macro lens. It looks like a regular lens, but you can get really close. And because you can get really close, you can get really high magnification. It's shell photography. Actually, I'm teaching a class on shell photography coming up, I think in November, and I think it's already in the website for registration, where I'll be exploring. And it's an old hobby of mine. I like to take photos, not only shells but birds and nature in general. So I hope that I can do a good job talking about shell photography at that course. And those lenses, they're not cheap. They're not impossible to buy if you're really into it. They sell for, I think, like 400, 500 bucks. So the equipment is not cheap, but you have other options, other brands that are less expensive.
I do have one question for Joe. Say on the Tampa drill, have they found a lot of them over at Punch Beach? Just curious.
No,
No,
not a lot.
just a few.
No. Yeah, the temper drill is one of those. I have several of those pet peeves going back. I've been, as some of you know, I've been here for quite a while in the museum. I come from the east coast of Florida, and when you move from there, I dealt a lot with the local Florida shells from the other side prior to coming here. But the two areas are very different. I mean, there are 50% only of overlap between the east coast and the west coast of Florida. So it's a whole new learning experience. And one of the things I learned right away was that we had a few temper drills in the collection, but they stopped at some point in time. And from that moment on, there was nothing. And no matter how much I did walk the beaches, and we, the museum, and the museum has a very strong backing of helpers and people who bring things on a regular basis. And, some of you are here in this audience, and no one was finding them. And just recently, like in the last year or so, a few of them showed up, and that's really something kind of cool. Not many. There are not many. If you go shelling in a lot of the people from the St. Pete, Clearwater Shell Clubs and Tampa Bay area in general, they do a lot of shelling along the Skyway Causeway. There's several islands there, rest stops, and it's really cool when the water is clear usually. And that species is very common there, but not here.
Thank you.
Okay. Thank you.
Thank you.